you know that today we have started the very important topic of your microbiology, and you know that we all already have discussed the features, classification, and definition of the viruses. Today we have started a very important topic of virology, which is structure and multiplication of TMV. So today we will be discussing the structure of the tobacco mosaic virus. You know that this tobacco mosaic virus is the first virus which was discovered in 1891 by Ivanovsky, and it is a phytovirus. Okay, it is a plant virus which is parasitic and pathogenic on the tobacco plant, and it causes a very well-known disease in the tobacco plant, and that disease is known as the tobacco mosaic disease. Now look at the whiteboard and try to understand the structure. Look at the whiteboard. Is the whiteboard visible? This is the structure of the tobacco mosaic virus. Look at the whiteboard. This is the tobacco mosaic virus structure. This is the structure of the tobacco mosaic virus. Look here. This is a cylindrical or rod-shaped virus, and it measures 330 nm in the length and 18 nm in the diameter and the width. Okay, and this virus appears like a rod cylinder, and from the one end of this TMV particle, and some part of the RNA, which is its genome, remains exposed. This has been in the red. RNA has been shown in the red ink in this diagram. So this is the RNA of the TMV, and all these grape-like structure which you find here sketched on the whiteboard, which constitute the structure of the TMV, are known as the capsomeres. Capsomeres are the subunits. Capsomeres are the subunits of this capsid. And what is capsid? Capsid is a proteinaceous sheath present around the central core of the RNA. So this tobacco mosaic virus consists of protein coat, which is known as the capsid, which forms the external covering sheath, and which surrounds the centrally located the single-stranded RNA. So this RNA has been shown in the red ink here, and all these capsomeres have been shown here in the black ink. This is the structure of the TMV. Now look here. I have told you that the capsid forms the covering external sheath. So this is the external sheath, and there is a central core. And in this central core is formed by the RNA, which is single stranded in case of TMV. So look here, this is the length 300 nm in the length, and 30 nm the part of the RNA which remains exposed constitute 30 nm. And in totality, the length of this tobacco mosaic virus particle is 330 nm, and the diameter of this tobacco mosaic virus is 14 nm. The wall, the thickness of the wall is 7 nm, and the central core is of the 4 nm. And you know that these capsomeres are arranged in the form of a ring. This is a ring of the capsomeres. Look, this is a very peculiar structure here. This is the one ring. This is the one ring of the capsomere, and one ring of the capsomere has 16.3 capsomeres. Look, I have written here 1, 2, 3, 4, 5, 6, and up to the 16. So the 16 capsomeres constitute one ring of the capsomeres, and in the central part of this ring there is a presence of the single-stranded RNA. And this is a very interesting to point out here that the RNA remains chemically interacted with these proteins. There is no superficial. There is not only the physical contact between the RNA and the protein, rather there is a chemical interaction between the RNA and the capsomeres, which is protein, which is polypeptide chain. Look here, this RNA, the RNA remains in chemical interaction with this protein part, which is capsid. This is the one ring of capsomere. I have written here one ring of capsomeres contain 16.3 capsomere, and three rings include 49 capsomere. So if you divide 49 by 3, you will be getting 16.3. So here 16 capsomeres have been shown present in one ring, and each capsomere here, and each capsomere, and one capsomere has been shown here, and each capsomere consists of a polypeptide chain consisting of 158 amino acids. So this is the structure of a TMV. 1 minute. Hello. So just I have shown you the diagram. So just you have seen the structure of the TMP. So the structure of, as far the structure of TMP is concerned, it is very simple, consisting of a central core, and the central core is made up of the single stranded RNA, and around this single stranded RNA, a large number of the large number of capsomere units are present, and each capsomere appears the shape of each capsomere is a grape like, and each capsomere consists of a polypeptide chain which has 158 amino acids. And the polypeptide chain acts as a protein, and polypeptide chain, what is polypeptide chain? A polypeptide chain is made up of a number of amino acids linked together by the peptide bond. So this polypeptide chain is the protein, and this polypeptide chain consists of a number of amino acids linked together by the peptide bond, which is CONH bond. And how many amino acids are present, and how many amino acids are present in the polypeptide chain which constitute the capsomere? The polypeptide chain which constitute one capsomere of a TMV particle has 158 amino acids. And how many capsomeres are present in a particular TMV particle? There are about 2130 30 amino capsomeres present in a single TMV particle. You know that there are 16 capsomeres in each ring, and one ring is present above another ring. Again, the third ring present on that ring. In this way, there is there are total 130 rings. How many rings are present? How many rings are present in a TMB particle? There are 130 rings in a complete TMB particle, and each ring of capsomere consists of 16 capsomere, and 3 rings of the capsomere include 49 capsomeres, and in totality there are 130 rings of capsomere. So if you multiply 130 rings into the 16 capsomere, you will be getting 2130 capsomere. So in a TMV particle there are 2130 plus minus capsomeres, but the number of rings are fixed. The number of rings is 130, and 3 rings, and for making 3 rings of capsomere there is requirement of 49 capsomeres. And if you divide this 49 by 3, you will be getting 16.3. What does it mean? It means that each ring has 16.3 capsomeres, and these capsomeres, which is made up of the polypeptide chain, remain chemically interacted with the RNA. And the RNA found in the TMP particle is a single stranded RNA, and there is chemical interaction. There is a chemical interaction between the RNA and the protein of the capsomere. So you can cite this. It is a very important example of the protein nucleic acid interaction. You know that the proteins interact with the nucleic acid. So this is the best example of the protein nucleic acid interaction, which is shown by the TMB particle. And so in this way, the structure of TMB is very simple. You will have, if you are asked to write in your examination the structure of TMB, if you are asked to write short notes of the TMB, at first you will have to draw the diagram which I have shown, which I have sketched on the whiteboard, and you must draw the structure of the TMB. Then you must show the rings of the capsomeres. You should also show the RNA present in the TMB particle, and then you must describe the structure. At first you will have to write that TMV is a rod-shaped phytovirus. Its length is 330 nm in length and 18 nm in diameter, and this is a RNA virus. and the protein constitute the external sheath, and the protein coat or the protein sheath is known as capsid, and capsid is made up of a number of subunits, and these subunits are known as capsomeres, and there are 130 rings of the capsomere, and each ring has 16.3 capsomeres, and the central part of the TMP particle has the single stranded RNA, and this single stranded RNA remains chemically interacted with the capsomeres, and and the RNA, which is single stranded in the TMP, is made up of about 6,300 ribonucleotides plus minus. There may be some variation in the numbers of ribonucleotide, but about 6,300 ribonucleotides constitute the single stranded RNA of the TMP, which makes the central core of the TMP particle, and the protein, and around this central core the protein coat is present, and the protein coat is made up of capsomeres, and total number of capsomeres found in the TMP in a TMP particle is about 2,130 plus minus. So in this way, today we have studied the structure of the TMP particle. In the next lecture, we will take up the multiplication of TMP, which is also very important. And you know that this TMP virus causes the well-known disease in the tobacco plant, and that disease is known as tobacco mosaic disease. So today we have studied the structure of TMP. In the next lecture, we will be studying the multiplication process of mechanism of multiplication. When you know that when virus attacks the living host cell, they multiply themselves inside the living host cell. You know that viruses are connecting link between the living and non-living. When the viruses remain outside the living host cell, they act as non-living, and when virus enters the living host cell, they act as living organism. And you know the most important characters of the living found in the virus is its capacity to reproduce. Virus, like other living organisms, have capacity to multiply itself, but not in the environment, but when they enter the living host cell. The virus can only multiply its number when it is present in the living host cell. So when the TMP virus attacks the tobacco plant cells, it multiplies there, and in the next lecture we will be studying the process of the multiplication of TMP inside the host cell. So now this lecture has come to an end. I will upload a note on the TMP structure today. So now this lecture has come to an end. That's all. Okay.